Hi, everyone, and welcome to episode 647 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and in this week's episode, we are excited to be rebroadcasting our interview with Dr. Louis Schipper, originally published on May 11th of 2015. Listeners, Louis works at the intersection of biology, geology, chemistry, and environmental sciences. His research investigates long-term changes in the organic matter in soil, nitrogen cycling and how we can remove excess nitrogen from agricultural ecosystems, and how carbon and other nutrients cycle both in agricultural and indigenous ecosystems. And Louis is dedicated to using science to develop practical solutions to real-world environmental problems. And in this conversation, Louis shares some outstanding stories about his life and his career, so get ready for this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am excited to be here today with Dr. Louis Schipper. So welcome to our show, Louis. How are you?
I'm great, thanks, Marie. Wonderful to be here.
Well, wonderful to have you here. And listeners, I'm excited to introduce you to Louis. And I'll have you know he is joining us today from New Zealand, where he's a professor in the School of Science at the University of Waikato. He received his undergraduate, master's, and PhD degrees in biology from the University of Waikato. And afterward, he accepted a postdoctoral position at the University of Florida before returning to New Zealand to work as a scientist for Landcare Research. Louis joined the faculty of the University of Waikato in 2005, and he is a fellow of the Soil Science Society of America, as well as a fellow of the New Zealand Soil Science Society. He is also an author on 2 patents. And listeners, today, Louis is here with us to tell us about his journey through life and science. So today, Louis, we want to get to know you, both as a scientist, but also as a person. So can you start off by telling us what you like to do when you're not doing science?
Oh, thanks, Marie. We've got a family, my wife and 2 kids. Kids are 14 and 18, and so they've really kept us occupied for about, the last 18 years, I guess. So we've always been very interested in the outdoors, and both the kids have gone through Cub Scouts all the way from about age 6 through to my son has just finished getting his Queen Scout, actually, which is the same as your Eagle Scout, I think.
Well, congratulations. That's awesome.
Oh, thank you. That's been great fun. And my wife and I have been involved as Cub Scout leaders for about 10 years now, I guess. My wife more than I at the beginning particularly. That means that we're often involved every week in some Cub activities, running camps, going on hikes and all that sort of stuff. So we do all that with the kids as well. So a lot of outdoor stuff, really. Not serious tramping or hiking, but a lot of day trips and walking. And we also have a lovely little house, which has a sort of what we would describe as a gully section behind it, like a little valley. It's almost like having a forest in your backyard sort of thing. And I spend time down in there trying to restore that native vegetation and other stuff in the garden. So that's kind of the sort of stuff that I'm interested in outside of my job.
Well, that sounds fantastic. It's great to hear a little bit about your family and your life and what you do when you're not hard at work. But next, Louis, we are going to chat about your scientific side. So can you tell us how you describe what you do to someone who might not be familiar with your area?
I'm really interested in how we might work with the land to minimize environmental impacts. So we as a society, a globe, use land in a whole range of ways, say for food production or for growing trees or whatever. And that's wonderful because it provides food and keeps people alive. And that's fabulous. But
it's pretty important, right?
Yeah, it is up there somewhere. Yeah.
Details.
Well, what is it? There's a fact. Did you know that it is the International Year of Soils as designated by the United Nations this year?
Really?
And that 95% of the food that we get around the world comes from soil. I don't know where the other 5% comes from, but 95%, the majority of the food that people eat in their lifetime is grown from soil. Now, that's marvelous. I'm not a production kind of scientist, but what does happen when we look to get that amount of food off the land, there can be unwanted environmental consequences, right? So you'll get nutrients leaving the land from fertilisers and from recycling. And that's a bit of a problem because it ends up in waterways and that can cause pollution there. Or some of it can be converted into greenhouse gases and go into the atmosphere and cause pollution there. What I am really deeply interested in is whether there are ways that we can work with the land while we're having production where we can minimise those losses. So decrease water pollution, decrease pollution into the atmosphere. I was trained as a microbiologist, so I look at what microorganisms are doing in soil, and this is a term called biogeochemistry, so it's sort of the cycling of nutrients in soil. And if we can understand how those processes occur in the soil, then maybe we can find ways of managing them so that we have the advantage of maintaining or increasing production while also decreasing unwanted effects on receiving environments like waterways or the atmosphere. So that would be the sort of central theme of the majority of the research that I do.
Well, fascinating research that you described, Louis, and I love how you put it into context that means something to everybody. 95% of our food comes from soil, so it's so important to understand how it's working and what we can do to make it work better. So fantastic. Now, we love to start off these interviews with a little piece of inspiration or a little piece of motivation for our listeners. I know myself, I'm always on the lookout for motivation to keep me going. So can you share with us today one of your favorite motivational or inspirational quotes?
Interesting. So I do have a quote, and it's a little bit messy, but I'll explain at the end if I could. So here it is. It's from A.A. Milne, Winnie the Pooh. When you are a bear of a very little brain and you think of things, you find sometimes that a thing which seemed very thingish inside you is quite different when it gets out into the open and has other people looking at it. Now, Winnie the Pooh is underestimating himself there a little bit because he had such a clear vision of his own world. But what he's talking about there is the very core of science, and this is the ability to have ideas and to test them. And you can have these great ideas inside your head, but unless you put them out there and test them against other people's viewpoints or test them experimentally, they're just floating around inside your head. And I've always thought, get lots of ideas, that's fine. And 90% of them, once I say them to somebody else, it's like, oh, that's actually not a very smart idea. And that's fine.
Sounded better in your head, right?
Yeah. But the challenge here for my students also, I say, look, you've got to put those ideas out there. Don't be afraid to be wrong, but test them. Put your ideas out there. And everybody says no such thing as a bad idea. The only thing about a bad idea is if you don't let it go. So if you put your idea out there, the thing that you want to test or try out, and you won't let it go when you realize that it's wrong. If you can let it go, then you can go on to another idea, and then eventually you'll find one that you go, oh, actually,
it's got potential. Yeah.
That seems reasonably interesting. Let's give this a crack. Let's try and figure out if this is reasonable or not. I mean, it's not a short, easy quote to remember, but it's one that I do kind of carry with me, if you see what I mean.
Well, I think that is a fantastic quote and a great application to science. And I really like this point you brought up about bad ideas, the one that you cling to, even though you've sort of shown that it's wrong. And I think that is so important in science, not to get attached to these hypotheses that you're forming, these ideas that you're having, and just sort of keep an open mind to look at the data and interpret it appropriately. So fantastic. Well, I'm curious to hear next, not only about what inspires you and motivates you, but about some of these people who've shaped your career. So Louis, can you talk to us about one of your role models?
I guess I've had a number of role models throughout my career, from former PhD supervisors, postdoc advisors, bosses at Landcare Research, and my colleagues here at University of Waikato. But I've often found that if I started to, in some ways, admire one person too much, you'd start to see their flaws as well. And that would always sort of wash away too much mentoring from them. So instead, what I've tended to try and do is pick from different people what I see their strengths as being. Like some of the most fantastic scientists I've met, my former postdoc supervisor, a very humble man. You know, he had a huge group, lots of publications, and yet a very quiet, humble man. And I've met a number of other scientists who have that same sort of humble nature, and I really admire that. I've met other people who work really hard. That's okay to a degree. I've met people who care for other students or scientists that they work with as well. So all of those aspects I've tried to pull from through time, but there hasn't only ever been sort of one sort of mentor that I've really clung to. As my career has developed, I've picked up different things from different people.
Definitely. I think you brought up some really good points there. And I think a lot of times it's hard to pinpoint one role model because there's not one person who you've met who you just want to be them. You know, a lot of times you do just sort of glean these traits that you admire. And I like these three that you've pointed out, kind of being humble, being hardworking, and sort of that caring and nurturing aspect of it as well. But I think you're right. As soon as you sort of get to know people better, there are a lot of traits that some people have that you're like, well, I don't exactly want to be like that. But
that's absolutely right. Yep.
Fantastic to hear about some of these people who've shaped your career. But I'm curious to hear next, Louis, about how your journey in science started. So can you take us all the way back to the beginning and tell us what got you started on this path?
Well, I guess I'm the first person from my family to go to university, but both of my parents deeply interested in science. So even as a child, I was really curious about the way the world worked. And then sort of fast forwarding, I went to university and wasn't quite sure what I wanted to do. Ended up doing biology and chemistry. I really enjoy biology, but when it gets a bit qualitative, I can't really understand it. But I found in microorganisms and microbiology a real structure in the way that biology behaves. They are in some ways much more predictable. So I felt comfortable in that. They're more almost chemistry sort of happening. So I did biology and chemistry. And then I finished my master's and I went on a bit of a trip over to the States and spent 3 months there. Came back to New Zealand, walked into one of the local research organisations who I knew, and I said, do you have any jobs available? And they had a 2-week position available. That grew into a 4-year PhD, including a year of working, looking at land treatment of wastes and how wetlands in the broader environment remove nutrients. I then got a postdoc at the University of Florida where I studied methane emissions from wetlands because wetlands are large emitters naturally of methane. I was there for a couple of years and then came back to New Zealand to take up a position, as you said, at Landcare Research, where I was an objective leader, leading research looking at the application of effluent onto land for recycling of that effluent to be captured in the plant materials. So again, you decrease the pollution of waterways. But I then got involved in a number of other projects as well, and my career then sort of took a whole different range of directions in terms of ways of working with biogeochemistry to minimise adverse impacts. And I was there at Landcare Research for about 12 years, I think it was. And then I just thought, well, maybe time for a bit of a change. Maybe I could try teaching at a university, and it just happened that University of Waikato advertised a position for a soil chemist. I wasn't a soil chemist, but close enough, they said. And so
they round up, right?
That's right. It's all how you look at it, really. And they just wanted somebody who could teach 2nd year and 3rd year soil science, and I have a graduate program running. So I thought, that sounds like a new, interesting challenge. I can see if I can teach. I can see if I can raise some graduate students and maybe develop a group. There was also a guy at the University of Waikato, a colleague of mine, Dave Campbell. He has some fascinating techniques for looking at ecosystem processes called eddy covariance. And this is a technique where essentially you can measure the movement of several small molecules like CO2 and water and methane and more recently nitrous oxide, which are all important greenhouse gases. And you can measure that movement out of the land and up into the atmosphere and back from the atmosphere down into the land. And you can do this over hectare kind of scales or acre kind of scales continuously at 10 times a second. And so you can get a really clear picture of what is happening from the land, the interaction between the atmosphere and the land at large scales continuously and very fast. Now, a lot of ecosystem scientists have been interested in this. But as a microbiologist and somebody who wanted to understand what microbes were doing in the land and not in a little bottle in the lab somewhere, I thought that this technique was just amazing and a real opportunity for science and for me to learn more about what microorganisms were actually doing. Not what we measure in the field, but what they actually do out on a farm at a large scale. So that was one of the attractions to come to the university. I thought, it wouldn't be bad being able to work with Dave and trying to figure out what is actually going on in the real world. And we've developed a very good working relationship over the last 15 years now, building up those kind of techniques and answering really interesting kind of questions. So now we have a nice big team of students, postdocs, and technical support to try and bring together his field of research and my field of research. And that's where I've gotten to so far, I guess.
Well, fantastic to hear about your career journey. And one thing that you mentioned that I really thought was fascinating was this 2-week position that you were able to turn into getting your PhD. So can you tell us a little bit more about that? You must have really impressed them in that first 2 weeks.
Yeah, it was interesting. I knew the people because I'd done a little bit of summer holiday work for them as a student. And unfortunately, one of their technicians was involved in an accident. So she was off for a couple of weeks and they had some pressing work that they needed to do. And I thought, well, here's my chance. I'm going to just work as hard as I can. I'm going to get involved. For some reason, I didn't seem terribly afraid to put my ideas forward, a bit like Winnie the Pooh. Mm-hmm. Put your ideas out there, work hard. I was looking for a PhD, but to be honest, my grades up until that point had not been that fantastic. They've been okay. I think a little bit differently than other people sometimes, and so it doesn't translate very well in exam situations. But when I got up into the research level side of things, I found that I could think and that I could have ideas and look at things in a way that other people didn't. And so I ended up working during that 2-week time period. It just extended a little bit as the technician was getting better. I ended up working with some other people in another part of the Forest Research Institute, as it was called at the time, doing a small contract job for them and bringing my microbiological skills to a bigger problem that they had about how effluent is treated in land. And so they needed somebody else. They needed another person. I just wandered down the corridor and I said, look, I'm happy to come work for you. Oh, I'd like to work for you if I could, please. But what I'd really like to do is a PhD. What do you think? And the guy said, well, yeah, sounds reasonable. And I think I ended up working 6 or 8 months for them as a kind of a contractor, if you like, and then transitioning towards a PhD. So I wasn't based at a university. I was based at a research organisation. It means that there's an incredible amount of focus on getting your work done quickly and efficiently. You also get exposed to a lot of different kind of sets of skills than you would necessarily at a university. So that's essentially how I turned 2 weeks into a PhD. Hard work and not being afraid to put my ideas forward in a quiet way. You don't need to shout from the rooftops, but just say what you think and back it up if you can.
Well, that is a marvelous story, Louis. Great to hear about how you made those opportunities happen because you knew what you wanted and you knew where you wanted to go. I think that is really inspiring for our listeners who might be in a position where their grades are all right, but they're not sure if they're going to make it into graduate school, just to kind of be persistent. And sometimes these backdoor ways into a PhD are great opportunities, and you just kind of have to line things up and be persistent, which is fantastic to hear about how it worked for you.
That's right. And I think this is the key thing for people to remember is that research is not just all about what the exams told you. It's about finding your strength. What are you really good at? Maybe you are good at maths, but it doesn't come out in exams, or interpreting the environment, or being able to come up with ideas, or being able to interpret data. Find that strength that you've got and build on it, work on it, and patch around it as best you can. I think that's critical for becoming a good scientist.
Absolutely. And I think there's such a wide variety of skills that are useful in different fields in science that there's really almost a place for any type of skill set, which is wonderful. But I think we've so far painted your journey through science to where you are today as sort of all sunshine and roses. We haven't yet talked about some of the struggles and the challenges, Louis. And I'd like to have you take us back to one of these points in your career where you were struggling with something. Maybe you had a major failure. Tell us what happened and how you got through it, so it might help some of our listeners out there going through the same thing.
I think everybody always comes up against things that are very difficult. Learning how to become a scientist is always going to be challenging, and your mistakes definitely teach you a heck of a lot. I was doing some work on something called a denitrification wall. Just imagine this. You have groundwater and it's moving towards a stream, and it's got nitrogen in it that's going to go into the stream, and you don't want that to happen. There's a process called denitrification that converts that nitrogen in the groundwater through to nitrogen gas, harmless nitrogen gas. So that's fabulous if you can get the microbial conditions right in the path of the groundwater. So I thought, I'll try this. I'll dig a trench and I'll take that soil and I'll mix it with sawdust or wood chips and I'll put that back in the hole. So now the groundwater is moving through this permeable wall of wood chips. And those wood chips provide an energy source to the microbes, and they then convert that nitrate in the groundwater through to nitrogen gas. So I built one of these things because it's a great idea until you test it and see what happens.
Of course.
And to be honest, the first one that I built is still functioning today, and that's like probably 18 years ago or something. It worked brilliant.
That's fantastic.
It was great fun. And then I thought, right, okay, well, that's a small scale one. Might have been 30 metres long or something like that. I'm going to build a really big one. I'm going to push some boundaries here and see what we can do in terms of really making these things hum, really work. So I built one that was about 120 metres long, and I varied the amount of wood chips in it, and it was 3 metres wide by 3 metres deep, and we had these big yellow machines in there digging the soil up and all the rest of it. Now, what was different about this site was into sand. It's a very sandy subsoil. So just imagine a sand dune that was wet. And we couldn't hold the sides of the walls of this trench up, and it would fill with water. So we decided, okay, tell you what we'll do is we'll mix the wood chip where it is. We'll just dig a hole, put some wood chips in, and then we'll use this bucket like a big spoon and stir it all up. That's how we'll get our wood chips in the groundwater. Mm-hmm. So that was great fun. And then we put wells, so things for sampling the groundwater, upslope of this denitrification wall and downslope of the denitrification wall, and some in the middle of the wall to see if nitrate was being removed, because you've got to test it, right?
Of course.
So the nitrate concentration upslope was 30 milligrams per litre nitrogen, which is quite high. And so it's okay, good, we've got our influent, that's excellent. And then in the wall, it was basically zero. So that was awesome. We were removing all that nitrate. It was doing as we thought. And then we went and looked at the well's downslope of the wall, and it was back to 30 milligrams per litre. And so there was something just completely wrong here. We had no idea really what was going on. Somehow this nitrate was coming into the front end of the permeable wall, then disappearing and then reappearing. That was like, oh, God, what have we done wrong here? So I was working with a hydrologist at the time, and so what we did was we looked at the rate of water flow through each of the sections of the aquifer. The short story is, when we mixed that sand underwater, it had packed down. So you imagine taking a bucket of sand without water in it, and that's a certain volume, and then you pour water on it and you give it a good shake. All the sand settles down, it compacts. And so what we had done was create a packed wall which wouldn't let water go through it. And so the water was coming in and hitting that wall at the upper end. It ducked down underneath this wall and popped up the other side. And that's why we saw high nitrate concentrations on either side of the wall.
Gotcha.
And low concentrations in the middle of the wall. So major failure? Absolutely. We're talking about a year and a half, 2 years to try and figure all this out and be certain that's what was going on. We published a couple of papers out on it, and those get reasonably well cited in terms of don't do this is not a smart idea. Mm-hmm. That was quite hard. I think one of the reasons I learned a lot from it, because I didn't attach to my results too strongly to begin with. I didn't think, oh, this didn't work, therefore I'm no good. It was more, oh, this didn't work. I wonder why that didn't work, and then went to try and figure it out. So lessons learned, don't build these denitrification walls into sandy aquifers. Go for the ones that have a stronger soil structure that can sort of stay open and don't compact like that. But we got through that.
Wonderful. I think you packed some excellent lessons into this story. And I really like this point that you brought up about getting those papers published that basically say, don't do this, because I think that is so important in science, but often really a challenge to do, to get those negative result papers out there, or failed methods types of papers. So great that you guys were able to do that.
No, that's right. And the way to get negative results published is to make sure that when you start out your experiment to begin with, that you anticipate, what if I get a negative result? And then look back at the chain of logic that has allowed you to establish that experiment in the first place. Because then you go, if I get this negative result, this suggests that this chain of logic that I use, that everybody else kind of believes in, is wrong. Then you write the paper about how that chain of logic must be wrong. So you use the negative result in a positive way to disentangle what people thought was their picture of the world and go, well, actually, that picture of the world is not quite right.
Definitely, and still have that compelling story. I like it. Well, Louis, we don't just want to talk about the tough times. Can you tell us next about one of your successes? And this can be a big success, like a grant or a paper, a project going well, or even just one of these little victories along the way.
Sure. One of the things that I've been really kind of proud of is actually a long-term effort that really stems from these denitrification walls. This is an idea that I had more or less at the same time as a guy in Canada, and the two of us have been working on that for, nearly 20 years now, I guess. And at the beginning, it was about putting wood chips into groundwater. And I talked to my colleagues about this approach, and they kind of look at me a little bit strangely. It's like, well, what are you doing, putting wood chips?
Right. You'll have Play-Doh next or something.
Yeah,
right?
That's right. Yeah, real. And a lot of it was kind of visualized as a little bit cute. So there was a long time of just sort of grinding away at trying to use this approach and developing new approaches. So we've now started, instead of putting the wood chips into groundwater, we now intercept tile drains. So they're drains that come from underneath agricultural fields that help remove the groundwater from underneath the croplands or whatever. And they can have high nitrates in them as well. And then what you do is you build imagine a swimming pool in the field. It's just a lined hole in the ground, really. And you fill it with wood chips and you pass the water from that drainage pipe into this wood chips and then draw it out from the other end. And you keep it flooded. And then the microorganisms, these denitrifying bacteria, convert that nitrate through to nitrogen gas. So we started looking at that approach. And then I would say about maybe 10 years ago, other people started getting in on this and started looking at this. And then most recently, we've started seeing farmers just installing them.
Oh, wow.
And we're starting to get better understanding of how these systems work. And I think we've got the basic principles down now. And now, particularly in the US, in the Midwest, the Mississippi Basin and around Chesapeake Bay area, there are a lot of people now doing research on these denitrification beds. How can we work with them? How can we put them in the ground? Do we cover them? Can you put crops on top of them? All these sorts of things. But these are sort of second order issues that we now have. We have a new technology that can be used in the right place, not everywhere, in the right place. And since we've published a lot of papers on this, I've worked with people in Rhode Island and all over the US trying to get these systems in place. And for me, that journey of some basic microbial ideas that we first observed in riparian wetlands, those little boggy zones next to streams, understanding how that works, what the microorganisms need to make that work, and then going, how do we artificially construct this into a field environment? Getting some papers published, and then getting buy-in from colleagues so that they then started doing work on these denitrifying bioreactors and other variants of that sort, has been a real pleasure, because for a long time it was viewed somewhat as kind of cute science, and now we've got symposiums on it or special issues of journals, and just a great group of people working at what are the limitations or advantages or disadvantages of these systems, where can they be used. And so that, for me, has been a real pleasure to be a part of that journey, if you like, from inception to now. Farmers going, oh, I can dig a hole and fill it with wood chips.
Well, Lewis, this is a fantastic story. And I think you brought up some really important points on being persistent. So can you tell us how you sort of stayed the course when people were maybe chuckling at your science or calling it cutesy? What kept you going and helped you get through to the point where you are today, where people are applying it and really respecting it?
I think I made sure that the science that I did was robust. And so I pretty quickly decided that here's an idea, we'll put it out there, and I'm going to test it at a field scale. I'm not going to see if it works at a lab scale, because we know that this works at a lab scale. If you add wood chips or a carbon source to a flask full of nitrate, you'll get denitrification. We know that. My question was almost immediately, let's go into the field and build a big one, and let's see if it'll work out in the real world. And I think even while people thought it was a little bit cute maybe to start with, they respected the science because the scale was right. And because I was asking the right kind of questions. And I was not afraid to show where I'd gone wrong. So that former example where groundwater went underneath the denitrification wall, people respect it when you go, well, it worked here, but it doesn't work here. Don't do this here.
Right.
Or you go, well, I think those denitrification walls, and I still believe this now, is that those walls are suitable in a very constrained set of conditions. And probably we won't see very many of those out there, and I wouldn't recommend them. But the denitrification beds, those big lined holes filled with wood chips that get water from agricultural drainage, they will work. And so if you're prepared, I think, to say, well, this works, here's my data, and this does work, and it is constrained in where it can be used, but it can be used in these kind of environments. If you are prepared to be totally honest with people in front of you, then you build credibility. Because it's not often the scientists will be upfront and say that they're incorrect or wrong, and here's the reasons why. But when people do that and they show why maybe they reached the logical position that they had gotten to, you garner respect and people will look more closely at your ideas. And then it's about finding some good colleagues, some people who will also listen and try your ideas. And usually if you've got a good idea, maybe a lot of people will kind of think it's a bit quirky or whatever, but really good scientists, the smart ones that are out there, will pick up on the strengths of the idea and go, oh yeah, okay, well, yep, I can see how that works.
It has merit, yeah.
Let's see if we can build on that. And then suddenly you get there.
Absolutely. Well, wonderful to hear how you took your ideas and really made them into something that can then be applied and that people are using to facilitate this process. I think that's fantastic. And we've talked a lot about the research that you've been doing over the course of your career. And I know every scientist keeps so busy with all the things that they love to do in science as well as outside of science. And it's hard to find balance. And I think one of the things that often goes by the wayside with the people that I've been chatting with is reading. And I know I myself am way behind where I want to be in terms of my stack of books to read, but I always love to add to it. So Louis, I'm going to ask you today to recommend a book for us, one that you've enjoyed, and it can be a science book or a non-science book.
I love to read. I don't read as much as I used to because of, I think, the amount of reading they do at work. I think one of the books, and this is definitely not a book for everybody, but there's a book called Neuromance, and it's written by a guy called William Gibson. It was written in about 1984. So remember that, 1984. Books need to be read in the context of their time, but this book, it picked up just all the major science fiction awards, but it really described what the web, the World Wide Web, could become in a sense in terms of integrated with our society. And it tells a story of a renegade guy who's plugged into the web, if you like. And so he, in some sense, a bit of a spy inside the web. It's kind of Matrixy-like sort of thing. And the first time I read it, I had no idea what was going on. The language, the jargon that the author was using was really hard to kind of get to grips with, but you could feel a good story. And remember, this is 1984, a long time before really the web has developed properly. So the vision of this book was quite remarkable. Since then, I've read it a couple of times more, and it's still very good. It kind of lost a bit of its polish perhaps because I've read it a couple of times now. But at the time, it was like, wow, this is near future visionary type material. So I think in terms of a fiction book, that would be one of my favorites. And then there's another one, Fermat's Final Theorem, is a sciencey book that was written by Simon Singh. And I don't know if you've come across this book, but it's the description of how a very old mathematical problem was finally solved by a mathematician in the US, and it took him like 5 years or 10 years. It's a very simple problem to explain, but very hard to solve. And he did some incredibly complex maths to get to solving this problem. Was seen as a huge breakthrough for mathematics. But the story behind, his name was Andrew Wiles, the story behind all of that was beautifully told by Simon Singh in this book called Fermat's Final Theorem. So if you ever want to read a book about the struggles of being a scientist, if you call a mathematician a scientist, I'm not quite sure, why not?
Certainly, yeah.
And then give some of the maths background, and I'm not a mathematician. That was great fun, read that.
Well, excellent recommendations for myself and our listeners, Louis. We'll definitely add those to our list online that we're keeping there. And I love to hear about the books that people are reading because I think it kind of shows a little bit about your personality as well. So this fascination with science fiction and also this sort of struggles of science and math and those sorts of things I think are really important to learn about as you're going through the process. So great recommendations. And we've talked a little bit throughout discussing your journey through science about some of the travel opportunities that you've had. So you mentioned you've come over to the States for a little 3-month kind of summer gig and then also for your postdoctoral experience. And I think scientists in general get to travel quite a bit. So can you think back on all the places that you've been for science and tell us about one of your favorites and what was it about this trip that made it so memorable for you?
When I was postdocing in Florida, I was looking at methane emissions from wetlands, and that meant that I needed to go out into wetlands, into the back areas of wetlands, and put chambers over plants to measure methane coming out of the wetland. And some of those wetlands in Florida and Georgia, Okefenokee Swamp, was just staggeringly beautiful. You get into a little boat and travel for an hour to get to your little spot, and there were snakes and alligators and insects and just staggeringly beautiful pitcher plants. I think I really enjoyed those trips. And then occasionally we were down in the Everglades once. We were sort of in the centre of 2 hours from anywhere sort of thing on this one of these metal boats with a big fan on the back. And we were sitting there making these methane measurements, and then thunderstorms started to roll through. So,
oh no.
Yeah, tell me about it. I'm sitting there. We're probably the highest thing for
just miles, right?
And it's a metal boat.
Oh, no.
So we jumped off the boat, and I'm from New Zealand, right? What do I know? I don't know anything about that. So we jumped off the boat and we hunkered down in the water. So now we're up to our necks in water, right? Just away from the boat, waiting for the thunderstorms to go by, and remembering that there are alligators here. And I was only probably 30 or something, so still pretty naive, really. Anyway, we ended up jumping back on the boat and just driving back through the thunderstorms because they kept coming. So that was memorable. And just getting into those wetlands was fantastic. And I do get to travel a fair amount. I go to the States probably once or twice a year, but I think it's the field trips, my ability to go out into wetlands, particularly where I do some of my other work, that is really quite special.
Well, that just sounds like an incredible experience and a sort of hair-raising story there, being stuck out there on a metal boat surrounded by water in the middle of a thunderstorm. Water and alligators even.
Yeah. They're more scared of you, they tell me, but I don't know about that.
I'm a little skeptical myself. Yeah. Well, thank you for sharing this fantastic story with us. And I think you're right. Some of these field opportunities that people get are just incredible, and being able to experience nature, to be out there with your colleagues, is just a wonderful perk about being in science in a lot of different fields. So in addition to talking about these travel opportunities, we like to talk about the people in science. And specifically, I think as you look back on your career, there are different groups that you've worked with, and each of them almost develops their own personality based on the people who were there. And I think there are stereotypes about scientists and what scientists are like. Often how they're portrayed in media perhaps does not always match real life. So we like to show the lighter side of science on our program. So Louis, can you tell us about one of your maybe funny science memories, or even just a quirky tradition that you've experienced in one of these places you worked?
Oh, right. So we've got a group of PhDs and postdocs, and we have regular lab meetings every Wednesday at 2. And the tradition now is that if you publish a paper or if you submit a thesis or whatever, then you have to bake a cake for the group. But it can't be any old cake. The cake has to be an abstract of the paper or the thesis that you've submitted. So it needs to reflect the research that you have done. And then I put them up on a website that I have called Research Cakes. So we're describing land research in a cake. And one of the success stories in this was that we have young students, and one of them, he just submitted his master's thesis, and he never baked a cake in his life. And so
really,
not only did he submit his thesis, not only did I mentor him to that, I got him to bake his first cake, and it was a really good chocolate cake. And he did an awesome job at describing his research on the top of this cake, where he had gone out and sampled dairy and sheep farms next door to each other for soil properties. And he had this cake with dairy cows and sheep on either side of a fence line with holes dug down into it. And he had gummy worms, because he dug up worms, and he had little M&Ms added around the place at the bottom for the stones at the bottom of the holes and all this sort of stuff. It's an abstract of your work. And so, it's pretty sciencey, but the cake is good. It's always gone at the end, and we make sure we have a photo of it. And I'm using that as kind of a record of what we have done as a group.
Well, I think that is phenomenal. I think everyone needs to put those pictures alongside on their CV with their respective papers.
There you go. Absolutely. That'd be a great one.
Well, I love this tradition. I love that it brings in the creativity and sort of forcing people to think about how they can portray or depict their research to people. I think that's wonderful. So how did this tradition start?
We had a couple of master's students, Anna and Alex, and they just decided one morning, I think they got up and they liked to bake, and they decided that they would make a cake, and then they got carried away. One of them is, well, they're both very creative, but one of them sort of decided that either side of the cake should represent both of their areas of research. So we ended up with a chocolate cake that had a denitrification bed on one side, so that's that wood chip thing, and the other side had an eddy covariance tower that I was explaining CO2 exchange. And over a wetland, we have some peat bogs out in the Waikato where I work, and so she had a little boardwalk where we have all our instruments, and they just brought that in, and I thought, this is a fabulous idea. And so I took a photo of it and thought, well, let's make this a tradition. And you sort of have to force it a little bit from the top, right, we're going to do this. And then I ended up making a couple of cakes as well just to get things rolling. And now I think...
Of course. Put in your support there. I like it.
Yeah. Well, I probably produced a third of them so far.
Gotcha.
I quite like it. It's quite fun. I'm no cook, but I can decorate. So I get in there and decorate cake. It's fun.
Well, wonderful, wonderful tradition. I love it. And I think it's such a great way to make the lab a fun place, to add that little piece of excitement, creativity, something for people to look forward to. So great tradition. And I think in addition to some of the work that you're doing in the lab, the people that you get to work with, one of the great things about science is being able to ask just exciting, creative, fun questions. And I think we've talked a little bit about the research that you're doing now, but we haven't yet talked about some of this work that you would love to do, some of these questions that are burning in your mind that you haven't yet been able to answer. So Louis, if I gave you everything, if things like funding and staff and technology and feasibility were not a problem, what is the one question that you would want to answer?
That's such a challenging thing. I get excited by a lot of different areas of science. One of the things that I've been doing recently is I've developed a collaboration with a molecular biologist. So this is a guy who works on enzyme scale issues. So he looks at enzymes in test tubes. And one of the big questions in enzymology is how they respond to increasing temperature. So as temperature goes up, how much faster do they go? And this is, strangely, a really important question in ecology as well, and at ecosystem scales. As temperature goes up globally, how is that system going to respond? And so we had a lot of coffees together and talked about this. And then Vic Arcus is my colleague. He developed a new theory which sort of made a fundamental advance on some of the basic understandings of temperature response of chemical systems. And he tested that on enzymes, so at the really tiny scale. And as we discussed things together, we found that we believe that this could also scale into soil systems and into ecosystem kind of scales. And that's what we've been doing. We've published a few papers on this so far, and it does look like the theory is going to hold. Getting funding for this kind of a fundamental aspect of research, what is the temperature response of biological systems that can scale from individual enzymes through to whole ecosystems, is challenging. Can we test this theory that Vic and I have been working on? We call it macromolecular rate theory. Will that scale across from enzymes to ecosystems? And then how can we use this? Even if you gave me all the money in the world, it would be interesting to try and design a research program that got at that. But I think it can be done, and it would be fabulous to see if we could have sort of this integrated theory of temperature dependence of biological systems.
Well, I really like this question. I really like that it addresses some of these issues of scalability of science. I think this is often one of the limitations that is listed in different manuscripts is, well, we don't know if this applies to everything. We don't know if this scales up. And I think this would really address some of these questions. And also just kind of touching on this interconnectedness between fields and how it sort of overlaps with ecology and a bunch of other different things. So it would have a pretty big impact, which is amazing.
We've had a lot of fun discussing this different societal meetings. Vic came to a soils meeting in the States with me, and he had a blast. He said, I've learnt more than anybody else here because I knew nothing when I first came here about this. And it took me and Vic probably 6 months to be able to start really properly communicating to each other because the difference in the language that we use across our fields. But it's been great fun, very challenging, but I think there is some real opportunity there in the future.
Well, wonderful to hear about one of these big questions that you've been thinking about. And I think when you work with different scientists over the course of your career, you not only get great ideas, you get inspiration, collaborations forming, but you also get some fantastic advice. So Louis, can you tell us today what you would consider the best piece of advice that you've ever received?
Oh, I think I've learned over time not to become attached to the results of any particular experiment that you do. I think that one thing that I see often with young scientists or students is that they design an experiment and they have a particular outcome in mind that they would like. And it doesn't work that way. And sometimes when that result comes out to be different from what you were hoping or expecting, you can feel kind of bad. You've got to step away from that. So detach yourself from the results that you expect in your experiment. It will give you a clearer mind when you get results that you weren't expecting so that you can reinterpret them in a way that allows you to make progress forward and not get stuck on, oh no, my experiment didn't work. It's, oh no, I didn't get the result that I was anticipating. What was wrong with my prior logic that allowed me to think that this would be the outcome? So detachment is important.
Well, great advice. And I think this is really important to not be setting yourself for disappointment or misinterpretation by having these expectations of what should happen necessarily with an experiment. So great piece of advice. Now, Louis, is there any other last piece of advice or maybe a last piece of inspiration that you want to share with our listeners today?
If I was going to say one thing, I think it would be educate young kids. If you've got 5 and 6-year-olds or even younger than that, they can learn science. If you are a scientist, see if you can help teachers, local teachers, because sometimes when they're teaching the young kids, 5, 6, 7-year-olds, they've got to teach them everything. They've got to teach them English and maths and science and all the rest of it. They can be a bit scared of doing science. Mm-hmm. They can be a bit scared of what science is all about because it can seem a little bit daunting. If you can work with those teachers, even if it's the teacher of your child or whoever, to break down some of the mysteries around science, do that, because it won't give you an immediate benefit, but in the long term, we will lift societal understandings of what science can do for society. But we need to work with children. We need to break down those barriers that science is hard and scary. And we've got to do that by getting rid of the jargon and explaining it to them as well as you can. Get involved, I would say, with primary school, we would call them here in New Zealand, primary school kids, so the younger kids. That's something that I feel very passionate about.
Well, Louis, I love this call to action for scientists. Get out there, start working on breaking down those barriers of learning science for young kids. So phenomenal note to end on here. Can you tell our listeners now how they can reach out to learn more about you or your research?
Sure. I have a website. That's www.waiber.com, and Waiber is spelt WAIBER.com. And you'll see our team there, and Research Cake is there as well. And we do take contributions. So if you've got a research cake out there that you'd like up on a website, you've got to give me a 50-word description and I'll see if I can load it up.
Perfect. Well, Louis, I'm going to have to get started on some research cakes here myself.
Well, that would be absolutely wonderful.
Excellent. I appreciate that. And Louis, thank you so much for joining us today and sharing your story.
Thanks very much, Maria. Really appreciate the opportunity to be able to share something that I feel quite so passionate about. And good luck to this podcast to keep getting science out to the community, because the more people understand about science, the greater the beauty they'll see. Thanks again.
Wonderful. Thank you, Louis. And thank you to our listeners out there. Join us next time for another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.